us via Zoom, but we really appreciate you coming in all the way.
Well, I have to tell you, like anything in the world, in life, especially in medicine and in business, when you actually see, the clinical situation or the technological apparatus, it's breathtaking what's going on here. So it's really impressive.
Yeah, it's interesting timing, and again, I mean, this is really an important milestone for us as a company, and we do appreciate you coming in here, Dr. Drew. As you mentioned, you and I connected a few months ago. We started talking about what Aditx is and what we do as a company, and to really share that with the audience one more time, we're focused on translating innovations and great sciences focused on immunology and advancing the health of our immune system into actual products that we can take to the marketplace.
So let me just drill into that a little bit. So I as a clinician can use some sort of, I don't want to say primitive, but some sort of gross parameters of the immune function. Do I have antibodies against coronavirus? Just sort of really what are very non-specific and sort of general ways of assessing things. Right. And to go beyond that in the past has required extremely expensive and time-consuming and labor-intensive sorts of evaluations, and not anymore.
Not anymore.
Yeah.
What we're looking at is, and without getting into the therapeutic side, which is really what led Aditix to the immune monitoring side, we're looking at objectively, with very high precision, being able to identify and quantify various analytes of our immune system. So we can provide an objective and meaningful status of what our immune system is.
And ultimately, maybe know whether or not we responded to vaccines, if we had coronavirus, if we have persistent immunity, are there things we can do to enhance our immune function? And then we get into a world of autoimmune disease and organ rejection or transplantation rejection, early intervention in juvenile diabetes, which is a great story I'm going to tell you about in a second. These are all what's coming.
That's exactly what our vision is for the company and the plans for our products and services.
Super exciting stuff. I'm going to hold you for a second because you and I are going to get deeper into the science, but I want to talk about Joe. Joe has sort of a way of framing this, because you come from such an extensive background in hospital-based, really, quaternary center hospital-based medicine. And you've been at the forefront of laboratory technologies, and this excited you. And when you described it to me, you said from the framework of the past, the present, and the future. And I like that framing. So tell us about that.
Yeah. Actually, first of all, I just like to say thank you for having me here. Thank you, Amro and Sharuk.
It's exciting to be part of this, right?
It truly is.
We're going to look back one day. You said this a couple hours ago, or an hour ago. We're going to look back one day and go, we sat here in November of 2020, or December now, I guess we are, and look at what happened. Look at how it becomes such an important clinical.
Yeah, you alluded to my background a little bit. I was director of cardiovascular laboratory medicine at the Mayo Clinic for a number of years, and in that role, I was involved with the many resources at Mayo of looking at multiple different technologies to develop tests that will assess cardiovascular disease, diabetes, and other related disorders like oxidation and inflammation that can occur with that. And so I was in the process of developing many tests in struggling with all the technologies. When I first met Amro and Sharuk about, again, a few months ago, they told me about the technology, and I realized that with this technology, there's applications beyond, even beyond immunity, but certainly encompassing immunity that go on to things like lipids and lipoprotein analysis, and making it easier to do a single lipoprotein analysis all in one assay looking at multiple lipoproteins.
Right. When I think about, for the average person listening to this, that one of the things that people need to kind of get their head around is that this is a technological breakthrough, and as such, it's giving us this window into all kinds of opportunities to deploy clinically that we're still kind of dreaming about, really. But some of the first applications are now here.
Yeah. In relation particularly to COVID-19 testing, which is what all of us are facing right now at this particular point in time, we like to think of it as, as you said, in the past, in the present, in the future. What happened in the past was everybody was getting the virus, and we wanted to know, do you have the virus or do you not have the virus? The present time right now is really about immunity, and that's the whole nature of Aditx itself. It's about immunity and the
am
testing
I immune to this virus or not?
Am I right? Have I been exposed to the virus, and have I developed immunity to the virus at this particular point in time? And what happens when that happens is antibodies will go up, and then over time, in 6 months or so, they'll go back down and they'll be level. And we still want to know after that if you're immune. So in the future.
So I'm going to stay with the present for a second.
Yeah.
So, and this is something that's in the press a lot, which is the antibodies are going away. They're no longer immune. That's not necessarily true. Because no one has had a widely available clinical assay to test that. Yep. No one has been able to challenge those assertions in the press, which have been grotesquely overstated. Yes, the antibodies always go up and go down, but that doesn't mean your immunity goes away.
That's exactly right. The antibody response is what's referred to as humoral immunity. And then in addition to that, there's a cellular immunity. So it's the cells like the T cells and the B cells that are responding to a particular virus. And once the antibodies go away, we maintain those cells to handle that. And so we'll be bringing, Aditx will be bringing forward in the future technologies that will look at cellular immunity as well.
Yeah, I'm very excited about that. And dovetailing into that will be, did the vaccine have some sort of sustained response?
Yeah. Or
is it like a flu vaccine where I'm going to have to take it on a regular basis?
Right. Yeah, first of all, when the vaccines become available, you're going to want to know, did you develop an immune response at all? Yeah. And then you're going to want to know, is it a long-term immune response?
Yeah. And my results. Turns out he's better than me, it turns out. So, Shruk, you're scientific perspective, because once you get into the weeds, it gets very complicated.
It does, and there are a lot of bits of information going around, and not all of it is very clearly understood. And that's because, again, we haven't had probably the best instruments to evaluate the immune system as a whole. We're looking at bits and pieces.
Sensitivity of our testing, and with this more, what should we call it, elaborate, or how should we characterize more complete testing, I guess, better way of putting it.
You're opening yourself up more to a false positive or negative, but if you're looking at a wider picture and casting a wider net to get a better look at the immune system, you get a much better understanding of what's happening in the immune system.
So we're going to keep this conversation amongst ourselves for about 20 minutes, but in about 20 more minutes, I want you all to, across the next 20 minutes, I want you to be sending in your questions. There's a Q&A function for those of you in the Zoom. It's just send those questions in. They're collating them here. They're going to ask them of us, and then we'll further discuss those questions. And by the way, you can follow the progress of this technology at Aditix, AditxTscore.com. So I think we should bring in the subjects now, shall we? And this is your chance to dissect our results. So what I have is AMRO and my own AditxScore, which are fascinating. They come with, before I review it, I want you to talk about the analysis that you get on the back of the score, which is very specific. You see this analysis on the back? It's a very specific set of evaluations and recommendations based on these score profiles. So talk to me about that.
Yeah, absolutely. So Aditix has developed what we call an interpretive algorithm for the testing itself. The interpretive algorithm basically looks at all of the different results here. And so we've got 3 different antibodies to 3 different antigens and the capability to do some neutralizing antibodies as well. Plus we can also test for the coronavirus itself with a PCR methodology. The algorithm basically takes all of that information for each individual. There are potentially 4,000 combinations of different results that are here, and we put all that information in it, and we have a specific comment that is related to each of those potential aberrations that's specific and very personalized to you.
And let me just say, the reason you need that is we're not saying antibody present, yes or no. They are talking about 3 specific antigen, the nuclear capsid protein, the S1, which is the spike protein, and the receptor binding domain, right? These are the
and Dr. Drew, maybe just to touch on what Joe just shared, because it's really important part of our platform, AditxScore, because if you look at AditxScore as a whole, it's a way to collect samples of some type. It's a way to process the samples, but we also must interpret the data
Yeah, of course.
into meaningful information
Yes,
to the, you know,
of
to the public or to doctors.
Yeah.
And the algorithm engines that Joe was touching on is key component of AditxtScore, because again, we get asked that question a lot. It's wonderful that we can tell you exactly what analytes we're identifying and the quantity of each analyte, but we must translate that into meaningful information. That's what we said day one. We want to position AditxScore in a way that provides meaningful information to the user or to the recipient.
So speaking of that, Taruk, let's go over my profile, which is the kind of, in a way, well, I'm disappointed, but it's the more interesting profile, because I have some antibody against the receptor binding domain, right? The RBD antigen. And had I simply been tested for antibodies or not, there's a probability it would have been yes. And yet, with the more complete score, you can see I have no immunity, no meaningful immunity against coronavirus.
Right, and again, looking at if we had just ran one test for you, typically how these tests work is you establish what's called a cutoff point, below which you're considered negative with respect to having antibodies, positive with respect to having antibodies if you're above that. So those cutoff points, and in your case in particular, 2 of those antibody isotypes against one of the commonly used antigens in the virus are right around the borderline. So it's difficult really to assess that. If we had just ran a test for you, a typical test to...
It's usually an IgG, right?
Yeah, typically an IgG.
I have IgG against the RBD, but
when you look at the whole panel and the view of how you've responded as a whole, then we can clearly say that you don't have immunity.
As much as that pisses me off. But yes, I don't. Keep going.
Right. And to add to that, one of the things that we started to do by being in the field of immune reprogramming, we understood that we must have tools to evaluate the responses. So here we will also be able to look at the cellular immune responses and then again get a much bigger picture and correlate that to
as well as our cytokine
antibodies.
system, right?
Right, and the point, and so the cytokines can be released by
and let me just emphasize again, these technologies have been out there to measure these things, but not like this, not with one test that's cost effective and clinically available.
Correct.
It would be extremely cumbersome. Joe, am I overstating this? And very expensive to do this old-fashioned way.
Yeah, you'd have to do them one at a time. You'd have to do 9 different analyses.
Right.
And have the cost associated with all these, where the technology.
And this is more accurate.
Yes, and this, the cellular assays that are currently available are not standardizable, so they cannot be turned into a clinical assay. So it's important to have a tool that can be used more widely for the population rather than just a research tool.
And you're still at Stanford, right?
No, I'm not.
Not anymore.
So I was at Loma Linda for many years.
Loma Linda, I beg your pardon.
Yes.
Loma Linda is where you're still doing this. Yeah. And our Stanford team I've spoken to, and the reason that occurred to me is that they're the ones that are so excited about this, when they see the potential of all this, because they've been laboring along the way you've described it, Joe, and all of a sudden there's this huge opportunity. The cytokine system will show us immunity or or propensity to infection.
So I think that the cytokine response will do probably at least 3 things for us. One, it'll provide some answers for some of the people who have essentially not become sick
Like
after.
And so really it's an answer why somebody could be an asymptomatic carrier.
Correct.
Also, will it have some import for the long haulers? Might we not see stuff?
It could. So then the cytokine profile, and by the cytokines, just to explain that, we mean basically proteins that are produced that cause a more severe inflammatory response.
People have, I think, heard of interleukins or,
Right, and certain interleukins are controlling the immune system and downregulating it. Some of them are upregulating it. So looking at those cytokine profiles can give us an understanding of why some people are convalescing well and recovering, some people are not. The other thing that we can get from the cytokine or the cellular immunity response is in the future be able to correlate a little bit more with those levels of the antibodies that are produced to see if we have more sensitive ways of detecting low levels of antibodies, how do those correlate with the presence of some of those memory cells that Joe was referring to.
I think we have questions already coming in, right? Is that why you're locked and loaded there? All right.
Yes, indeed. Someone has asked, if someone is shown to have COVID-19 immunity, can I skip the vaccine?
I think that's, everyone's looking at me for that one because it does fall to clinicians to make that determination. You should not make that determination on your own. You should discuss that with your doctor for sure. Had I personally, I could tell you how I would approach it for myself or even my family. If I had clear-cut immunity, like AMRO does, which we'll discuss, Sharuk, if you could get us into that, and neutralizing antibodies, I would forego the vaccine, or I'd certainly have my kids forego the vaccine. But that doesn't mean that they'll never need the vaccine, by the way. So it's something that needs to be monitored now. We may be able to answer that question when the cellular immunity is more clearly operationalized, but that's what I would do. By the way, I'm in a hurry to get the vaccine, especially based on my profile now. So, but talk about AMRO's results as it pertains to...
Maybe just Dr. Drew,
Yeah.
you know, certainly because we kind of expected that question to be asked.
Yeah.
And from our point of view, we certainly not going to make that recommendation. I mean,
Right,
like
you wouldn't be making
that
those.
they would need to discuss.
This is not
But
a
it's-
substitute for your physician.
it's
This is-
not-
This is something for your physician to use to make that
Exactly.
determination.
We want to provide information, at least as we see it, and then they need to make that determination with the physician.
Right, and by the way, that's an ice pick moment in the now. Whether or not that applies next year, we don't know yet. So, but AMRO's results were
So,
quite different than mine.
I think AMRO's, talking about AMRO's results, and again, going back to what AMRO actually was saying earlier today, was, you know, the focus has been so much on the infectious or the infection status of individuals, and not so much on the immunity status of
Right.
individuals. And his test results, for instance, in comparison to yours, really highlights that. And as we move deeper at is what you know, how's the population responding to this virus? And it's very interesting also, and we hear this all the time. He doesn't recall being sick. Now, he does remember, you know, at times or earlier in the year having some symptoms, but there are many people who may have flu-like symptoms, and they automatically think, oh, I had flu-like symptoms in January, I must have had COVID. And it turns out that probably the vast majority of them didn't. So there are a large population that may not have had any exposure to the virus at some point, and with a profile like what AMRO is showing, he is clearly showing high levels of antibodies and several different antibody isotypes to based on those levels of antibodies, we can say that he has been exposed at some point and has had an immune response that could be protective.
And you have seen some, I don't know if this is a topic for real discussion or not, but there's some relationship with blood types and this sort of response?
So there have been some reports, earlier reports, that certain blood types infection. Whether that is the case or not is yet to be studied further. But what again is exciting about what we are doing is having the tools to essentially evaluate the immune system and look at various parameters and take different cohorts and look at why someone may be asymptomatic, maybe someone has antibodies but never had symptoms and therefore never really became sick, and what might be the various individual parameters, blood type, HLA type, individual differences among different people.
Joe, I'm wondering when I look at Amro's profile, the fact that, so again, we're talking about these three different antigens, which is the nuclear capsid protein, the S1, and the receptor binding domain, right, the RBD.
Correct.
Amro does not have much response to the S1, which I think is isn't that usually what they're measuring in the more commonly available assays?
Yeah, sometimes they'll measure the S1, sometimes, more frequently, probably they do measure the RBD.
RBD.
And
So what I'm wondering is whether this would have been missed by a standard assay.
Yeah, it's a possibility if they did an S1 or they had antibodies for one particular antigen
Yeah,
that
so
we
this
may
is
have.
the part I want people to get.
We
is that
may have actually missed.
Yeah,
missed a
the
result.
sensitivity and specificity of what's out there is pretty sort of gross. It's sort of not specific. It's not
Yeah.
very accurate. I want to use these words properly, but I guess accuracy would be the right word.
Yep,
Yeah.
that's right. And the nice thing about this is even more comprehensive. Amro did develop antibodies to the RBD, both IgG and IgA, but he also developed antibodies to the nucleocapsid. So the RBD is part of the spike protein. We didn't see S1, which is also part of the spike protein, which may mean that there is an opportunity for certain antibodies to bind one site and maybe block that other site. And so if you're testing for that other site in a single assay, you'll miss that altogether. The nucleocapsid antigen is, so the spike and the RBD are on the surface of the virus. The nucleocapsid is internal. So it potentially may mean, and this has to be borne out with science, but it potentially may mean a more robust response to the.
Well, that's what I just, out of curiosity, I'm wondering if the NP antibodies are more associated with virus neutralization.
Yeah, it's a great question, and I think that
You would
one
think
of the
so,
things,
right? Just
the
in terms of
neutralization, it tends to be that the RBD site is associated with the ACE gene, that it allows.
That's how it gets in.
Right, exactly, and so it may be that the RBD site is more associated with neutralizing antibodies because of the fact that that's the mechanism whereby the virus enters the cells. It may be that this response that goes, clearly has not only a response to the surface, but also the inner portion of the virus itself.
Right.
Could mean that there's a
NP.
more robust response, and those RBD assays, antibodies are the ones that are going to.
It also makes me wonder, and this is just spitballing here, whether or not there are people that maybe have a profile like Amro's that are really fighting off the virus as it's attacking, and that's what creates the asymptomatic states.
That could be that virus never really fully gets hold.
Fully gets in because they get these RBD antibodies up front fast, and maybe there's a cytokine piece of that too, right?
Right, and that'll be
Interesting to know.
The other thing that will be very useful with this type of panel, again, we've currently put 3 antigens on this, but we can expand that significantly.
Well, that's what I want people to understand. I guess we'll sort of get into in a few minutes, sort of dreaming about the future and how all you can. Right. This is the crazy breakthrough, is you can expand in all kinds of ways, right?
Yeah. Well, we're still on this topic. If I can just add one
Please.
more thing, that Amro maybe thinks that he may have had some symptoms in the past. We don't know if that was associated with this response, but we had a group of 10 people in here to do testing, and we found that 2 individuals, Amro and another, had no previous symptoms.
Did they have the same profile, same pattern?
But they had actually a little bit different pattern. One of them had IgG and IgM rather than IgG and IgA, into some different antigens, so a different pattern. But it an indication that people with no symptoms truly can have developed immunity. So one additional case, and this one's really clear cut. We had an employee who was exposed to 2 close friends, and he was exposed to the virus. They both got sick, one of them pretty severely sick and the other a more mild case. We told him to isolate, so we did, don't come in here and spread this to other people potentially, and so he did. And we tested him for the antigen early on, or for the virus itself early on, and it was negative. We didn't get to test him again for another about 10 or 11 days, and it was still negative. We had already tested him for antibodies in the beginning, and there were no antibodies present. And he developed a really strong immune response. He never had a single symptom. So he's no symptoms at all, a strong immune response, developed immunity to SARS-CoV-2. We saw the whole picture. We were able to track it.
Right, so that's what we all want to be, right?
Right.
We all want to be him.
How many people are out there that are worried about developing, coronavirus that maybe have already been exposed to the virus
Absolutely.
and developed an immune response and potentially are immune?
Or even, from my perspective, a little scarier, gets tested as antibody positive and is not really immune. That's,
Correct.
And goes about their business and gets something. But let's not forget to talk about sort of, again, the future. We're going to get a question in a second. They're locked and loaded, but about how we can enhance our immune function and measure that, and what the sort of the future holds. So, all right, question.
How does AditxtScore differ from other monoclonal antibody tests being released to determine immune responsiveness to COVID-19?
True.
So again, the AditxtScore is designed to look at the immunity status. That's the main thing. And we're not looking at any one parameter that can be missed or subject to false positives and negatives based on a particular test. It's really designed to look at the immune system as a whole and various components of it. The other thing that is very important is we have strive to provide meaningful information to patients or people who are tested. And that comes with the algorithm that has been developed that Joe referenced earlier, and those algorithms there's a lot of work that has gone into those in order to provide information that is understandable and usable by individuals who are tested.
It occurs to me that I probably didn't give you full and adequate introduction, because when people hear about a lipid lab and stuff, they sort of understand what that is. Describe to people what your background is in science, just to, if you can, a couple sentences, because we're leaning on you for all the science for a reason.
So I started in microbiology and molecular genetics, and my background was in studying host pathogen interactions for many years, focusing more on the pathogen side for probably the first half of my career in science, and now focused more on the immunology side or the host response. And that's where we have now been developing technologies that are focused on not blindfolding the immune system, but reprogramming them to become tolerant to a particular...
Things like organ transplantation, autoimmune disease, that sort of thing.
Correct.
Did we answer that question adequately, do you think? How Aditix is different than other?
Yeah, I believe so, in multiple ways.
Okay. So let's talk about the future. So let's start to get into that. Where this is going. And again, this is like the very, very beginning.
Right.
This is where we can start this whole process.
Dr. Drew, I think by looking at the presence, we're able to really paint the picture where AditxScore can be in the future. Because look at what's happening with COVID-19. Especially when it started in December, January, February, and look at the, confusion that we've all experienced. Positive, negative, PCR, antibodies, symptomatic, asymptomatic, right? And we've all made decisions and policy decisions, and rightly so, because we had to act fast. But reality is, what we desperately need is information. So the purpose of this discussion, the spirit of this discussion, is for us to share with the world what we're working on. And, as we get closer to launching AditxScore, of course, we'll make the appropriate claims. That said, we're looking at AditxScore to become an informational tool to provide to various stakeholders the needed information to make decisions, whether they're physicians, policymakers, so on and so forth. And if you look at AditxScore, and we repeated this theme over and over again, the ability to identify and quantify with high precision multiple components of the immune system, so we can come back and say, here is the information, right? So somebody can make an informed decision. Let's take that concept and begin to paint the picture for the future. So COVID-19, great example, we're right in the middle of it. As we get into vaccination and post-vaccinations, we believe the question is not only whether I'm infected or not. That's something that can be done. That's a commodity. Am I really vaccinated? Am I really immune? And we can provide, again, the appropriate information for someone to make that decision. But take it beyond infectious diseases, because COVID-19 is not going to be it's not the first and certainly not going to be the last. infectious diseases as a whole, we will need an immune monitoring service like the one that we're doing with AditxtScore. It's no longer a reactive action. We look at it as a proactive process where we need to monitor our immune system. Take it beyond infectious diseases. What about susceptibility to certain diseases? Can we, and that's really a question, can we predict particular diseases in the future based on your immune profile?
You're thinking diabetes.
T1D comes to mind, certainly. Type 1 diabetes comes to mind. That's one area that we're looking at. Look at potentially organ rejection. Can we predict a pattern of our immune system where it's starting to reject a particular organ? And the list goes on and on. So that really comes back full circle to personalized medicine, because it's not one size fits all, and I think we can all agree. And what's really needed is accurate information, or again, not to characterize accuracy, we want to make sure we're very careful and appropriate as far as regulatory is concerned, but we at least need to provide information as far as the immune system, where somebody can make a decision based on our information and other information sources. And that's where we're taking it.
Does it sound familiar?
Yeah. I think
Just like the lipid thing, right?
Yeah,
very much.
Can I just comment on,
Yeah.
the type 1 diabetes is an autoimmune disorder. And, I was cardiometabolic disease, which means cardiovascular disease and diabetes at the Mayo Clinic. We really were looking for autoimmunity. Is this diabetes case? Now there's latent onset immune diabetes. And that's happens in older adults where they actually have an autoimmune component. So we developed one assay for autoimmunity to the pancreatic beta cells associated with diabetes. They have the ability to, and it's an expensive assay, I would just say that.
Right.
They have the ability to look at 4, and if more are developed, because 4 are on the radar as being potentially clinically useful, in one assay with less expensive reagents to get that done, which is really nice.
But the way Amro describes the future in terms of immune monitoring reminds me of the way we think about lipids and monitoring.
Yeah.
And all the different people, again, people think in sort of large clump terms about lipids, when in fact you can really begin to break that into very definable biologies.
Absolutely. We typically will measure total cholesterol and LDL cholesterol to determine our immunity. There's a lot of data which supports that really it's not the cholesterol that's in these particles, but it's the lipoprotein particles themselves, including LDL, VLDL, HDL, and IDL. And then there's another lipoprotein called Lp(a). This technology, as I looked at it, and this is what I got so excited about, is I've been working on nuclear magnetic resonance, electrophoresis, immunoelectrophoresis, and other ways of looking at the lipoproteins, all complex and difficult to do. But this technology potentially has the ability to allow us to look at the various lipoproteins and quantitate all of those lipoproteins, which are really the driving factor behind the buildup of atherosclerosis or plaques in the
again, back
arteries.
to Amro's point about the personalized approach, each of our biologies, metabolic biologies we're talking about now, are so different that the more specific you can get, the more personalized, the more specific you can get with your therapeutics and preventative interventions. My head's spinning a little bit about what can I do to enhance my immune function? Are we going to be making make recommendations, but is that the kind of thing we could monitor?
We want to be objective about it, right? So as we provide information, again, we want to provide objective information, because as you know, I mean, we all
I take my zinc, take my vitamin D,
exactly,
I take my vitamin C.
But
am I really doing anything?
But you would, exactly. That's really the point where we're going. We want to be able to show it in numbers, by the numbers, and here's where you are, here's where you were, and here's where you're going.
Interesting. Question?
What is the regulatory pathway for AditxScore? Do you need emergency use authorization?
That's a great question. I know that we've been getting that question quite a bit. Right now what we're doing is we are looking to commercialize this AditxScore for COVID-19 as a lab-developed test. And that's why we announced recently that we established our interim operations here in Richmond, Virginia, and we secured CLIA certification. In parallel, we're also pursuing emergency use authorization, which we filed in August. And that, again, is a parallel thread that we are pursuing. Our goal with that is, eventually when we, if and when we get approval or authorization, that it will be very helpful input in 510K, because ultimately we want to be able to secure 510K for AditxScore and the various assays we're looking to launch. So we're doing both in parallel, as a lab developed test, as well as an EUA, and eventually a 510K.
And is the EUA in a very specific, narrow aspect of the result, or is it just the technology that's getting the EUA and the applications then can be a little broader?
For now, we actually filed specifically for AditxtScore for COVID-19, and that's
Got
what
it.
we
So
started with.
that's what you and I have had.
That's correct.
This test.
Yeah.
Okay.
This one is as a lab developed test, what we're doing right now.
And this
That's
is
part of our,
what the emergency use authorization is for.
correct. That's what we filed, yes, and we have not received an authorization yet.
So I haven't heard your sort of dreams about the future. Where do you see this going? What do you hope we get to in terms of the personalized application?
How often do we hear, I had a close friend, he was in great health, had no issues, and just had a heart attack out of nowhere. We are really looking at shifting from reactive medicine into a proactive way of monitoring our health. So rather than waiting basically waiting until you have a symptom,
Until you get sick.
till you're sick to go to a doctor and then getting prescribed tests that may or may not hit the mark, we're looking at a continuous way of evaluating your health. And I think that will also give incentive to people to follow it. So if I'm told by my physician, exercise, eat well, you say, okay, yeah, fine. I may do it for a week after I see him and really not do much afterwards. But if I have a way of really monitoring and measuring a measurable change, then I'll be motivated because I will start seeing differences from when I started. So that's where we see this as monitoring going, and then you will still have the need for therapeutics, and again with the therapeutics, where we are going with that is to be more targeted, and in the case of immune-mediated disorders like organ transplantation, autoimmunity, or allergies, being able to be more targeted and not blindfold the immune system.
Yeah, it's interesting. We're all sort of coming at it from the really kind of the infectious disease sort of point of view, but I imagine the allergy community must be very excited about this kind of thing.
Right.
And if you don't have allergies, you don't realize how problematic they are for people.
Right, and they're the flip side of the coin, right?
Yeah.
So,
In terms of overactivity of the immune system, yes.
It'll be interesting to see also what the allergy prognosis is after all this, basically being homebound and not exposed too much. But these types of platforms, whether it's on the monitoring side or the therapeutic side, have tremendous applications for all of these.
So I'm trying to read between the lines of what you're saying. Are you imagining a day when we will be taking people that have hyperimmune reactivity and having targeted exposure therapies? Not necessarily desensitization therapies, but sort of rather than hiding at home from all the stimulants, meaning the immune stimulants, you go out and expose yourself.
So the exposure that that we get naturally, of course, gives us bits and pieces, and that sort of builds a healthy immune response.
That's our theory, yeah.
But when we are doing sort of artificial desensitization procedures with, let's say, a particular allergen, the problem is you don't know how the body is going to react. You're not really giving it the proper signal to become tolerant to that allergen. And that's where our therapy comes in, and the approaches that others are looking at is to have what we would consider a tolerance delivery system, so that you're delivering that allergen in a way that the body looks at it and says, hey, I'm going to lay off of this thing and not be reactive to it.
And is that by using the Aditix system to specify which antigen they should be exposed to, or is it adjusting the immune system?
So that's a great question. I do believe that the AditxtScore platform can be used for identification of targets. But then on the reprogramming side, we're using technologies to teach the immune system to become tolerant to those particular
Can you give an example
targets.
of that so people can
So,
understand what you're talking about?
A perfect example, again, going back to the type 1 diabetes example that has been brought up, that's one disease that has been fairly well characterized as far as what the targets are in the pancreatic cells where
Beta
insulin is being produced.
Which is, it's, by the way, historically so funny to me because when I was in medical school, the idea that it was an infectious or autoimmune disease was outrageous. It just
Right.
it was being raised at the time, and people were like, "Tuh." And that's
Yeah,
how-
yeah. So the because there are basically 4 antigens in the pancreas that have been identified as a source of attack, immune attack. By incorporating one or more of those targets in a tolerizing approach, the goal then is to induce tolerance to that particular antigen so the immune system stops attacking it.
Right.
And again, the earlier you can do it, the better, and that's where the complete story with what we're doing with Aditix is having a monitoring system where you can identify the patients early.
And the specific antigen.
And,
right?
and the
So
antigen.
early and where it's attacking.
Treat them, then monitor them during treatment, and then also have a way of monitoring them later so that you're not waiting for them to become symptomatic again, but you can look at whether they're developing those antibodies to those antigens and then maybe provide them with follow-on treatment.
So that warrants revivifying. I want to state it again because it's so extraordinary to think about this as near-term future, which is essentially elimination of type 1 diabetes, right? This is the type of diabetes when you're a child, your pancreatic cells stop producing insulin, you have to take insulin. It's because of an immune attack on these cells, and with this system, you can measure what's being attacked, create tolerance, the attack stops, and then monitor to make sure there's no further future attack, no diabetes, no insulin, no nothing, none of the complications, just a normal child from then on, essentially, by blocking that immune response.
Right,
Yep.
right.
Question. And we're in preclinical, just to...
Yeah, this is all fantasy. This is all
We're in
fantasy.
preclinical.
This is where we want it to go. This is the fantasy for the future.
Exactly.
Yeah.
Yeah, if I can just expand on that a little bit, this is, every time we talk, I get more excited about
Yeah,
this. My background isn't in immunology, it's in cardiovascular diseases, and cardiovascular disease is a multi-complex disease. There are many potential causes, and the reason that you might have cardiovascular disease is different than the reason, the root cause for my disease.
But embedded in what you just said, though, is this big story we're telling here.
Yep.
Amro puts it under the heading of personalized medicine.
Yep.
I put on under the we're all very different biologically and genetically, and then require personalized medicine to be able to approach in a accurate way.
Yeah. What's really interesting to me is whatever the root cause is, many times there's an immune response to that root cause. And so we may be able to, even earlier than we can, if we have an analyte that we've developed to try to identify that root cause, we may actually see an immune response to that root cause prior to us being able to determine that root cause even
Give me an example.
exists.
I kind of see where you're going. Do you have something in mind?
Yeah, like for instance, if your root cause is say even if it's related to autoimmunity, you have an autoimmune disease.
Where'd that come from?
Right, where did it come from? And you've now created a problem where you're developing atherosclerosis, and it might be because of, I said there's many potential. Homocysteine, could be Lp(a), lots of different analytes that we're used to looking at and monitoring, but that process has started, and your body's made an immune response to it. And this, I'll go to the next, and we may be potentially able to detect that early on. But in addition, if we don't detect it and we get to the point where we've developed cardiovascular disease, well, your immune system is functioning to try to fight that off.
Oh,
and you've got this innate immunity that is, not well. I mean, really is maybe potentially draining your immune system's ability to react to something like COVID. And that's why we see, people who have heart disease, diabetes, and others maybe are more susceptible to COVID because their immune system is already fighting off.
Right, you're talking about this sort of thing, this thing that we just sort of sweepingly call inflammation.
Looking into the future,
yeah, we just go there more.
We know,
and we know that with the metabolic syndrome, right?
We know that we can look at a number of these potential informational things that are telling us that we've got an immune response. Can we characterize it to say, yeah, that immune response is because this patient is developing atherosclerosis?
Right.
And can we find those signatures? The technology, and this is future thinking.
Yeah. But then if you find that
there.
and it's in the middle of a pandemic, you better address it because it
Absolutely.
increases the risks of the
Yeah.
outbreak of the infection. And my fantasies go towards cancer and stuff too, because I've spent a lot of time, particularly working in prostate cancer, and we do a lot of thinking about how the cancer is able to evade the immune system.
Yeah.
Now I know we're not specifically working there with AditxtScore, but this technology, maybe, I don't know, am I getting way into the off the grid here with this, or do you ever fantasize that there could be some? We probably haven't even thought of yet what the applications would be.
And you know, one application that comes to mind is identifying which patients might respond better to a specific treatment.
In terms of things that open the immune system back up to attacking the cancer, in terms of those kinds of technology.
Right.
The
And-
CD4 does.
And that truly enhances the efficaciousness or efficacy of a particular approach, because now you have selected the right cohort.
And this is back to the personalized, approach. Question.
How does someone get their AditxtScore, and when do you anticipate it being available?
So we're looking to launch January of next year, and that's our goal right now. And AditxtScore.com will have the information, and you'll be able to add your name to the mailing list that we have. AditxtScore.com. AditxtScore.com.
Yeah, let me say it again. AditxtScore, one word.com. And you can, and if you don't sign up, you can just follow along. The news will be there about how this technology is progressing.
Can I go back just a minute?
Absolutely,
There was a
Joe,
question about the EUA, and you mentioned an LDT, or a laboratory developed test. And part of the reason that we're going to be launching in January is because there's some stringent criteria for developing a laboratory developed test. So you have to go through a number of validation steps to just make sure that the assay is performing correctly in the lab that you have. And
Define
then,
what you mean by laboratory developed test.
yeah, it's a test that that is developed in a particular laboratory that's CAP or CLIA certified. And we have to
opposed to research laboratory.
Yeah, a research laboratory. So this means that you now can test this on patients, okay, and it's available for patients. And in the CLIA and CAP, the College of American Pathologists, are the bodies that regulate the laboratories and the laboratory developed tests that are set up. They have stringent criteria in terms of what we have to do to make a test that's available to use in clinical practice. So we're launching, we're going for the emergency use authorization, but in the laboratory Aditix here, we're launching with a laboratory developed test that has undergone the scrutiny that is necessary to pass CLIA and CAP standards.
And you come from more the research world. I mean, how different is that?
We've been developing a lot of products for the clinic. So going through regulatory process is something that is part of research, because we work really on applied research and clinically relevant products.
Question.
How is AditxtScore different from Abbott Labs quick test?
The PCR quick test?
Yeah.
So if we're talking about the PCR quick test,
I think that's what they mean, the Abbott machine.
Yeah.
PCR quick test.
If it's a PCR quick test, it's really measuring presence of the virus. What we are measuring is the immunity status to the virus.
You gave us a PCR too, though.
We did, so AditxtScore does include both the infectivity status and the immunity status.
So what I would say in terms of that question is the quick test, and this is not a quick test, this is the standard PCR, right?
Correct.
Yeah, is a good test. It's a good test, but that's, do you have the virus now? Right. As opposed to your overall immune status. Very, very different questions, different assessment.
But the results that are provided by the quick test are provided.
Right, the same result, just more of a traditional method of delivery.
That plus more.
Plus a lot more.
And that's an important piece of information. And we see it as important to actually still understand whether or not someone is
producing virus.
Exactly.
And again, just to state something, that producing virus on a PCR doesn't necessarily mean infectious or infected, but it's an important part of the status and where somebody is in the evolution of this illness. Next question.
Who do you anticipate will be using this test? Epidemiologists, clinicians, or both?
We believe both because it's back really to the information that we provide to the users and others as well. I mean, we're also talking about, speaking about the future, think about companies and biotech companies that are developing therapeutics, and they need information about the immune system, various aspects of the immune systems as they develop, as they administer, and post-administration of their drugs. So we see multiple applications for AditxtScore.
Now this lab, one of the function of this lab was to do some very fancy lipoprotein analysis and gut analysis, that kind of stuff. Was that always ordered by a clinician? Because this reminds me of the same thing. I personally, as a patient, would be interested in a more evolved, more in-depth analysis of my immune function. Does that can I initiate that? I'm wondering in your experience
in the past. Now
in this current lab.
Yeah.
Could a patient initiate it, or did a clinician have to do it?
We've always in the past had clinician initiated. We've talked a lot about the possibility of going direct to consumer. Always good to have clinician oversight in anything that you do go to direct to consumer, but that is a possibility for the future. But previously, always clinician initiated analysis.
Got it.
If I can answer on the epidemiology point, just
Please.
so really we want to provide a service to the population to really help us understand many disease processes, COVID-19 first, and then we'll look at other diseases. The because of the wealth of knowledge that is developed by the AditxtScore, the epidemiologists are going to be very interested in the research and how this applies to populations. So that's more of a population-based study. So there's really going to be tremendous opportunities to do studies that we can publish in peer-reviewed scientific literature, opening up more information about how immunology and the immune system interacts with multiple disease states.
I would imagine your peers are very excited about this.
Yes, and one more area I think that has a real utility for this platform is if you're developing vaccines, because if you're developing vaccines and you're targeting specific antigens for that vaccine to be more direct and make sure that you're eliciting immune response to the antigen that you want, not an antigen you don't want. Again, this platform can include an array of different antigens from that target virus and really facilitate development of vaccines as well.
Yeah, you were talking to me off mic about newer approaches to vaccine technologies that was surprising to me. And again, my head goes to cancer and things like that, where there's vaccine therapies may one day have huge application, right?
They could, and again, it's all immune related. So if you want to measure that response, whether you're dealing with an infectious disease and cancer on one side, or whether you're dealing with,
you know what, it's interesting. I feel like we're at I just think about an automotive sort of analogy. Like right now we've got like we have a Plymouth from 1930 and trying to imagine a Ferrari in 2020, and it's hard to, but we know we're going to get there. You know what I mean? We can see the future. Next question.
Are you going to develop a test for long-term memory B cells?
That's our goal, certainly part of our plans.
That's soon, yes?
Yes.
Yeah, and memory T and helper T cells and the whole, all of that humoral immunity, cellular immunity, is coming.
Yes.
I want that test. I really want that test. Because, and by the way, again, the epidemiologists are going to be really interested in that as well.
Absolutely.
So, well, this has been a very interesting conversation. Are there more questions coming in? Are we, okay, all right, bring them on over here. As we...
Apparently some indication that individuals with type O positive blood are more resistant to COVID infection than other blood types.
So we sort of addressed this earlier on, but I'll let you say it again. And there's kind of, it's a complicated story. I know that in the New England Journal article, they had 6 different genetic loci that they associated with resistance to infection. One of those happened to be associated with an O blood type, right?
Right, right. And the AditxtScore can then look at the various or the patient backgrounds and correlate that with whether their disease state, what that was, or how they may respond to treatment as well, or even vaccination. So there are certain segments of the population that may not respond to a vaccine. It would be very, very useful to know after a vaccination protocol whether you have actually developed immunity or not.
That's a really interesting thought. Now that you talk about that, we could on our request form, if the patient knows or the doctor knows the patient's ABO blood type, we could just ask for them to fill that in. And so if we got that information, that really opens the door for epidemiologic studies as well. And yes, we got that information, but we could also take that information, incorporate it into the interpretive algorithm, and then adjust some of the responses based on your blood type if it bears out that there's real importance. And I think more research needs to be done.
Right. When you build your algorithms, are you pulling research? Is it?
Oh, absolutely.
Yeah. And so you're
yeah, we
basing
look
it in
at
some
all
sort of
the currently
evidence-based.
available publications
Right.
and what they're showing us. It's all evidence-based.
Right.
And we try to make, so in any test that we do, we want it to be actionable. We want it to identify a particular disease state and something that we can have an action for. And so you'll see in the interpretive comments, it gives you ideas based on not only your laboratory results, but what are your clinical symptoms at this time. These are the types of things that you should be doing to make sure that everything is going to go well for you in the future.
Here comes another question.
Where can people go for more information?
That is AditxtScore, AditxT score, one word, SCORE, AditxtScore.com. You can sign up there. You can watch the news as it unfolds, and we encourage you to do so. It's exciting stuff, and I'm just privileged to be a part of it. And I thank you for asking me to be here today. I'm so glad I came to Richmond to see Joe, to meet you, and to see what's going on in your laboratory, and just a great team, Shrok. I mean, the scientists I've spoken to from Northern California, there's something that always sticks in my head that one of the scientists said is, we can do anything with this. We can do so much. And so it's really about our imagination and then our ability to prove the results. Anything you want to say, Amber, in closing?
We really appreciate your involvement. We're pretty excited about our mission.
Yeah. Joe, last words?
Yeah, it truly is a team approach. So many people involved in getting this done.
Good team. Shruk?
It's a team that is deeply rooted in science, but they want to do things that haven't been done before.
Right, and that can change people's lives and help doctors, which is what I'm excited about. So again, we thank you for being here. It's been a privilege to share some thoughts this morning. And again, it's AditxTscore.com to follow along. Hopefully we'll do, in 6 months, we'll do another one and talk about the cellular immunity and get updates from the laboratory. And this is an interim lab, right? We're going to be there's going to be another lab we'll probably be doing this from next time as things move along. So we thank you for being here, and I'll see you next time.